students. In this video lecture, we are going to discuss synaptic transmission. Okay. Synaptic transmission means how now impulses are transmitted from one neuron to another. That is, synapses is the space between two axons of neurons. Okay. Before that, we are going to discuss the conduction of nerve impulses through myelinated nerve fiber. You know what is myelinated nerve fibers? Usually, nerve fibers which are covered by myelin sheath are called myelinated nerve fibers, and there are two types of nerve fibers: myelinated nerve fibers and non-myelinated nerve fibers. Conduction of nerve impulses through myelinated nerve fibers is called saltatory conduction. Saltatory means it is jumping. Impulses travel through myelinated nerve fibers as in a state of jumping. Okay, it is not a usual transmission. It usually jumps from one side to another. Okay, and it is in the form of nerve impulses, in which impulses jump from one side to another. Okay, you know, in myelinated nerve fibers there is some split in the covering which is called a node of Ranvier. So other areas are insulated because of this presence of myelin sheath, and only node of Ranvier is the area which impulses can potential difference can create. So conduction of it's because the nerve potential jumps from one node to another. That means one node of node of Ranvier to another node of Ranvier, instead of traveling through entire nerve fiber. Okay, so this kind of transaction or transfer of impulses are called saltatory conduction. In usual nerve fibers, it travels from one side to another. It's a continuous traveling of impulses, but in myelinated sheath, it jumps from one node of Ranvier to another. So the conduction is called saltatory conduction, and it is around 50 times faster than non-myelinated nerve fibers. Myelin sheath act as an electrical insulator, so only from uncovered area this impulses potential differences can be created. That is, only node of Ranvier is excitable and the action potential is generatable only in the node of Ranvier. So from one node of Ranvier, the impulses jump into the another and next. Likewise, the travels means impulse travels in such a way, so the conduction of impulses are called saltatory conduction. And in this picture, you can understand the difference between these two. In first picture, you can see in green color, you can see the myelin sheath and a gap in between these myelin sheath, which is called a node of Ranvier. Okay, this all these myelin sheath act as an insulator, so the action potential cannot be created in this area. Only this area, that is in node of Ranvier, this are excitable. So impulses jumps from one node of Ranvier to the another. So this type of conduction is called saltatory conduction or jumping. In non-myelinated nerve fibers, in second picture you can see it travels continuously. The impulse or potential difference create excitable in all the points, so it travels continuously. Okay. Next, we will discuss what is a synapses. I already told you this is the junction between two neurons. Okay, so synapses is the functional junction between two neurons. If it is between two neurons, it is called a neuronal synapses. And there are another type of synapse, that means between a muscle and a neuron, and which is called myoneural synapse or a neuromuscular junction. So two types of neuronal synapses is there. Between two neurons, it is called a neuronal synapse, and if it is in between a muscle and neuron, it is called myoneuronal synapses. In a synapses, in a synapses, we can see that the two axons are present, and it is called a presynaptic terminal, and then the another one is called a postsynaptic neuron. Presynaptic neuron and post synaptic neurons. The impulses are traveled from presynaptic to postsynaptic neuron, and the gap between these two is called a synaptic junction or synapses. Different types of neuronal synapses. Axodendritic synapses. Here the synapses is between axon endings of presynaptic neuron and dendrites of the postsynaptic neuron. That is, here the junction is in between an axon and a dendron. The presynaptic end is a axon and the postsynaptic neuron end is a dendron. So in between an axon and a dendron there is a synapses. Then it is called axodendritic synapses. Then second, it is axosomatic synaptic neuron, that is synapses between axon endings of presynaptic neuron and cyton of postsynaptic neuron. That is, it is the junction between axon and a cyton. Cyton, you know, it is the cell body of the neuron. Sometimes there may be some synapses which is in between a neuron, in between an axon and a cell body. Then it is called a axosomatic synaptic neuron. And second, it is axonic synapses. That means the synapses between axon ending of a presynaptic neuron and axon ending of another postsynaptic neuron. Here the synapses is in between two axons, that is a presynaptic axon and a postsynaptic axon. So that kind of synapses is called a axonic synapses. Okay, the axon endings are generally a knob-like structure which are called a synaptic knob or terminal knob. A synaptic knob is separated from the postsynaptic neuron by a fluid-filled gap, which is called a synaptic cleft. In this picture, you can see the synaptic cleft. And in now endings of this axon, you can see many vesicles which are filled with some chemical called neurotransmitter. And during impulses transmission, some changes occur and they elicit this neurotransmitter into the junction, and which are received by the receptors present in the postsynaptic neuron. Synaptic knob is packed with mitochondria, smooth endoplasmic reticulum, microfilaments, a group of secretory bodies which are called synaptic vesicle. They are secretory bodies. That means vesicles inside the something is present that is called a neurotransmitter. So these secretory bodies are called synaptic vesicles. Synaptosomes released a transmitter substance which is called a neurotransmitter to the synaptic cleft, which brings about the transmission of impulses across the synapse. Across the synapse mean in between two synapses there is a junction, and how the impulses are transmitted through this junction. This is by the help of a chemical which is called a neurotransmitter. So neurotransmitter is the chemical which helps the transmission of impulses from a presynaptic neuron to postsynaptic neuron through this junction. The synaptic cleft has bounded by presynaptic membrane which releases neurotransmitter, and it is received by another membrane which is called a postsynaptic membrane, and they have receptors for receiving this neurotransmitter which is released by the presynaptic neuron. Three types of synapses are there: chemical synapses, electrical synapses, and conjoined synapses. Chemical synapses means here the transmission of impulses occur in the form of chemicals. These chemicals are called neurotransmitters. And electrical synapses, here the transmission of impulse occur as electrical impulse. If such a transmission should occur, then there should be a close contact between post and presynaptic neuron membrane. Then only the electrical synapses is possible if these two membranes are in close contact. And conjoined synapses, here the transmission of impulse occurs by electrically as well as chemically. Both ways are possible in conjoined synapses. And synaptic transmission. Electrical transmission usually occurs in tight junctions. You know what is tight junction? You may already studied in cell biology. This membrane modification, tight junction. Here the two membranes very close to each other. So synaptic membranes are so close to each other. So impulses transmit as electrical potentials. Chemical transmission, when action potential or impulses reaches in the synaptic dome, the membrane become more permeable to calcium ion. Okay, here in chemical transmission, the impulse from one membrane to another is transmitted as chemical signals. How it is transmitted? So when the action potential reaches in the synaptic knob, it releases the calcium ions. In the presence of calcium ions, certain enzyme become active and which activates synaptic vesicle and which releases neurotransmitter. From there, this neurotransmitter attaches to the receptors which are present on the postsynaptic neuron. Then depolarization occur in the postsynaptic neuron. How this depolarization occurs? Because due to this, the sodium channel starts to opening, which cause starting of the action potential, that is depolarization. If the neurotransmitter is inhibitor, if it is excitatory, there are two types of neurotransmitters: excitatory neurotransmitter and inhibitory neurotransmitter. If it is excitatory neurotransmitter, which due to that the sodium channels become opens and which cause depolarization. Okay, if it is inhibitory neurotransmitter, which cause opening of the potassium channels, so repolarization occurs. Excitatory neurotransmitter make membrane potential less negative, which excite the postsynaptic membrane. Sodium channels are opened and it elicits postsynaptic depolarization. It increases permeability of the sodium to the postsynaptic membrane, and it may be cholinergic or adrenergic. That means cholinergic, acetylcholine and adrenaline are the two type of excitatory neurotransmitter. They always elicit an impulse, or they cause depolarization. Okay, they excite membrane. An inhibitory neurotransmitter, it make membrane potential more negative, inhibits postsynaptic depolarization. It is done by increasing the permeability of the potassium ion instead of sodium, so it causes postsynaptic membrane to become repolarized, and so it become more negative. So GABA and endorphin are example for inhibitory neurotransmitter. All these neurotransmitters are used in psychological treatment. For treatment, we are usually using these kind of inhibitory or excitatory neurotransmitters as medicines. And in this, you can see how this excitatory synapses and what is the difference between this excitatory synapses and inhibitory synapses. It is K plus, sorry, it is not Cl minus, it is K plus, and here it is sodium, and one cause depolarization and another one cause depolarization. Okay, you correct it as K plus. Neurotransmitter, I already told you these are the chemicals which helps for the chemical transmission of impulses from a presynaptic nerve endings to a postsynaptic nerve ending. Acetylcholine is a kind of neurotransmitter. It's a cholinergic neurotransmitter, and it is released by neurons outside the CNS as well as some neurons inside the brain and the spinal cord. It increases the permeability of sodium ions. That means it is excitatory and which cause excitement or which generate action potential by causing depolarization and cholinesterase. I mean, when it releases the impulses starts, it excites, and how it is stopped? It's stopped by the action of another enzyme which is called cholinesterase, which inactivates these acetylcholine after some time. Otherwise, the continuous excitation occurs, which is prevented by the another enzyme which is called cholinesterase. And here it is given some excitatory neurotransmitters: glutamate, norepinephrine, dopamine, acetylcholine. These are excitatories. Then inhibitory neurotransmitters are the GABA, gamma aminobutyric acid, serotonin, dopamine. These are the example for inhibitory. And I already told you these chemicals are usually used in psychology as medicines for treating many defect, mental disorders. Aminobutyric acid. It is an inhibitory neurotransmitter. It brings about hyperpolarization in the postsynaptic membrane, and it is secreted by cerebral cortex, cerebellum, basal ganglia, retina, etc. Dopamine. It is an inhibitory neurotransmitter, and it is produced from basal ganglion, hypothalamus, limbic system, neurocortex